Let's get rolling. New topic, alleles. This is usually part of a tiny little bit of a chapter in the early part of the genetics textbook, not the course manual, but a typical hardbound textbook. But this is a really important concept for the rest of the discussions we're going to have in class, and then beyond this class into cell biology and evolution and other classes that many of you are going to wind up taking, especially if you're a biology major, evolution in particular. So please finish up the Socrative activities. We'll talk about that in about a half an hour, so you've got time. Oh, quick point, grade distribution from the exam. Bell curve, but more A's, which I like. I prefer everybody to get an A if possible. So on Friday, everybody will get their exams back. There's still one student left taking a makeup exam, so I'm not giving out any exam information yet other than everything's graded. I just can't give you the details. All right, so what's an allele? Does anybody have a simple definition, given what you've read for this chapter? What's an allele? Doesn't have to be a formal definition. What does it mean? What sort of a molecule does an allele have to do with? Yeah, I heard somebody whispered something, sounded right. Genes. Yeah, DNA. It's a genetics class. So let's start off with, we're talking about alleles, have something to do with genes. So we're going to be talking about double helices, my favorite molecule. Your favorite molecule too, I'm sure. What's an allele? Let's say this is chromosome 1 from your dad, chromosome 1 paternal, chromosome 1 from your mom. Right, each of you presumably have a mother and a father, or did at some point, and you got your genes from them. So, is your version of chromosome 1 you got from your dad exactly the same as, in DNA sequence, the chromosome 1 you got from your mom? Are your mom and your dad identical twins?
I should hope not.
It would be kind of hard to have identical twins of different sexes that could have had you, right? Identical twins are usually the same sex. So the probability that your two chromosomes, the one you got from your dad and the one you got from your mom, is 0. They're going to be different, even though they're the same chromosome. What's the word that we use for the same chromosome from the two different parents?
Homologous.
The H word, homologous.
Homolog.
These are homologs. That is, they're both the same chromosome. They have the same genes. I'm going to use these little right-handed arrows to indicate the positions of genes, just randomly drawing on the chromosome. They've got the same set of genes. They're in the same positions on the same chromosomes. If we banded the chromosomes, they would have the same banding patterns. So they're homologous, 2 copies of chromosome 1, but they're different. We can make up differences. So there could be an A at one specific spot on one homolog on the chromosome you got from your dad. There could be a C on your mom's version of the same chromosome. And we've talked about that. So those distinctions, what do we call these? Place where it's different one chromosome to the other. It's a mutation. In this case, it's a single mutation, which we also call SNP, single nucleotide polymorphism, SNP, single nucleotide polymorphism. Somebody's got a difference. Either your dad's mutant or your mom's mutant, or they're both mutant. Can't tell. SNPs. So that makes these 2 alleles, 2 versions. So if you want to use a plain language word instead of allele, just use version. These are 2 versions of the same chromosome. There's the paternal allele, there's the maternal allele. They're mostly the same, but at places that there are differences, like this one spot on the chromosome, there's a SNP, then those are two versions, 2 alleles. Here's a different example, more concrete example perhaps. We've got human chromosome 11. So two different versions of human chromosome 11. On this chromosome is the hemoglobin B chain gene, HBB. What does hemoglobin do? The molecule, the protein, hemoglobin? Yeah.
Yeah, it binds to oxygen and carries it throughout the blood.
Okay, so it's the protein with iron, once it binds iron, binds oxygen and carries it through your blood and delivers oxygen to your tissues. It's a gene that's on the left end, it's near the end of chromosome 11. So each of us presumably has 2 copies of chromosome 11 and 2 copies of the HBB gene. And then you can have a mutation in the hemoglobin B gene that is the sickle-cell mutation. There's usually one specific mutation in that gene that causes sickle cell anemia, causes your hemoglobin to fold improperly, to be a different shape, doesn't bind oxygen as well, it also clogs capillaries and does other things. So those are 2 alleles, 2 different versions of the HBB gene. They differ based on a mutation. A take-home point then is that for something to be called an allele, there has to be a genetic difference. That's how we define versions of genes, different sequences. So the sequence of HBB on top, which we're going to call, what do we normally call the term for the usual or the typical version of a gene? We don't call it mutant, we call it the opposite, usually. It's like original, wild type. And I asked you to watch a couple of topics ago, I think, actually. I didn't remember to talk about it in class then, but it's a good time to bring this up now. What's that National Geographic, is it National Geographic video I asked you to watch? Who's typical or something like that? The most typical person? Some of you maybe remember watching that. Who's the most typical person?
One is Chinese person.
Han Chinese male with no bank account and some other stuff, right? The purpose of having you watch that video was to think about the concept of wild type, or what's common, what's frequent, what's typical. That's what it was, the most typical person. So in the sort of vague point here is that we call the allele, this is all allele language, we call the allele that's wild type the one that's most common, the one that's found in most people. In other words, if everybody on the planet had the sickle cell mutation, we wouldn't call that the mutation, we would call that the wild type version because most people would have it. So it's all definitions and semantics. What you call wild type and what you call mutant, it's relative. Wild type is just whatever is the most common version of a gene. Any questions or concerns up to this point? Okay. So if we're going to talk about alleles, we're going to start getting a little bit into the genetic nomenclature, which was the questions at the start of class that I asked about genotypes and haplotypes. So geneticists have a particular structure, a format that they use to describe how alleles are related to individuals and to each other. So in this case, does anybody know what symbol geneticists tend to use to represent wild type? Instead of writing wild type every time, we use a plus sometimes. So sometimes, just for shorthand, wild type is abbreviated plus. And likewise, sometimes mutations are annotated either as asterisks or as minuses. Just shorthand. So the common one would be plus, the rare allele version of a gene would be minus. And let me invent another gene here, gene B, just for the sake of education, that's on the same chromosome, so also chromosome 11. And there are going to be 2 versions of this. There's going to be, we're going to call B minus the one that's on the top strand, and B plus, or the wild type version of B, on the bottom chromosome. Not strand, chromosomes. So each of these are double helices, as usual. So genotypes. What is the genotype of this individual at the HBB gene? This individual, both these chromosomes, same individual, same cell, same person. How many wild-type versions of the gene HBB do they have? One. How many wild-type versions? One. We've all got 2 sets of every chromosome, so we all have 2 alleles of every gene. This person has 1 plus and 1 minus. To make this a genetic annotation of a genotype, that is a genotype, description of the genetic composition of a person at whatever gene or spot on a chromosome we're talking about. What's the genotype of this individual at HBB? They're plus, minus, 1 plus, 1 minus, 1 wild type, 1 mutant version. The only thing we have to do to complete this is add a slash. You've probably seen this before in intro bio courses, genetics in high school. Maybe you've seen it, maybe you haven't. The slash indicates the separation of the homologs. In other words, everything that's on one side of a slash, the left side, say, is the genetic composition of one homolog, one of the chromosomes. So on the top chromosome, they've got the wild type version of HBB, it's plus. On the other homolog, slash, there's a minus, so we put the minus on the other side of the slash. So there's a slash that separates every set of homologs. 2 homologs, 1 slash. So what's the genotype? Here's your turn. What's the genotype of gene B? How would you write that? Please do write it, and then tell me what you wrote. Gene B. So what's the genotype of gene B? What are the 2 alleles?
-/+.
There's a minus allele and there's a plus allele. Okay, so I've heard minus slash plus. Does anybody agree or disagree? Or have any questions about that?
That's what I put also.
Okay, we've got a second vote. Now the rest of you vote, please. Especially those of you in the back. Do you have any questions about this at this point? Do you always have to put the plus first? No. You can put them in either order. It doesn't matter. Minus plus, minus slash plus, plus slash minus, fine. If you're this type of genotype, there are 2 types of genotypes. Now we're just doing vocabulary. What are the 2 different words we use to describe genotypes? There's one where you've got 2 different alleles. That's H something zygous. There's heterozygotes, and then if this individual is plus over plus or minus over minus, they would be homozygotes. So these are both heterozygous genotypes. Hetero means different. They have 2 different alleles, heterozygous. If you've got plus over plus or minus over minus, that's homozygous, same alleles, homo. So we've got 5 really important vocabulary items at this point. Alleles, heterozygotes, homozygotes, wild type, mutant. That's about as tough as it gets this chapter. Now it's all just using this information. Any questions? All right. Let's see. So you're diploid, think of yourself as a 2-car garage. You've got 2 copies of every chromosome, so you've got 2 alleles possible, maximum, of any gene. You can't have more than 2 versions of a gene in your cell because you're diploid. 2 copies of every chromosome, 2 versions of every gene. They could be the same, homozygous, they could be different. You're heterozygous. That's all of the options. When is this not true? In what sort of a situation is it not true that an organism would have just two versions of a gene?
Polyploidy or lymphloidy, I think it was called.
Yeah. So if you were a salmon, for example, and you were tetraploid, you'd have 4 copies of every chromosome. You could have up to 4 different versions of a gene in your cell, in every cell. Up to 4, no more than 4. So you're limited by the number of chromosomes you have. That's good news, because we really only have to worry about 2 alleles at a time for most cases in most organisms we might study, unless you move into strawberries, other plants, salmon, and some other kind of crazy animals. Last time we talked, hopefully this will be a little bit more engaging than me telling you about vocabulary, asked you a question. Should we be able to genetically test people for their alleles, that is, which versions of genes they carry, to enable you or somebody to decide who you could have children with? And most of you probably said, well, let's see what the results were. 5 said absolutely yes. Sounds reasonable. Probably 22, not sure. In the middle, only 6. So it was pretty polarized. It was basically half the class said, yeah, probably, and half the class said, probably not. So the rest of the next few slides are just a quick story, something I saw recently in the news. And this is the headline that caught my eye, because I'm a geneticist, so I thought, hey, I better read this. Right? Those crafty news people always come up with catchy titles that make you want to read more. So I did. And of course, you can read along with me. I'm not going to read this verbatim. But basically, some of these first quotes on the first couple of slides here, and you can definitely go look at this article yourself if you want to, says, yeah, it's a pretty common thing in Nigeria to have this be like the first or second question you ask somebody on a date. What's your genotype? Like, what do you do for a living? What's your genotype? What's your major? What classes are you in? That sort of thing. So it comes up at the top of the list. And the reason for this is sickle-cell mutations, that they're really prevalent in Nigeria, in other places too, but especially in Nigeria. And so it turns out that it's a really important conversation to have when you're starting to date somebody, because if you think you might want to have kids someday, you probably shouldn't marry somebody that has the sickle cell gene if you also have that allele. Because if you both have the allele, the version of sickle cell that's mutant, then they could be combined in your kid, and your kid could have sickle cell anemia. And so it says things like, in May, the state parliament in Nigeria's eastern region passed a bill making genotype testing compulsory before marriage. This is what prompted me to ask, is it okay to have genotyping before you decide who to have kids with? Well, in some parts of some countries, apparently now it's the law that you should or need to. Yeah.
It was actually pretty common practice, or actually I think mandated in different regions of the U.S. previously, that you had to get blood testing done before you got married.
Fair enough. So there were, yes, in different parts of the U.S. at different times, and right, I'm not sure currently if this is still the situation. Basically, a primitive version of ancestry testing, how closely related you were to somebody, could be used to, you could do a blood test and decide if you were able to get married to somebody that you wanted to get married to. And probably for some other traits as well, I think, not just.
Make sure that the fetus would survive pregnancy in the first place.
Right. Yep. So I'd like to do a quick Google Classroom exercise with you, a little bit of calculation. So if you'll open Google Classroom, you should all have this new exercise there. It's another Google Sheet. So I'm going to read you a couple statistics. They're up here on this slide. And then what I'd like you to do is to fill in the yellow spots on the Google Sheet that you have. This is not for points. You don't have to turn it in. It's practice. You can turn it in if you want feedback on it. But this is practice. So I'd like you to do this, give you the instructions, and then I'll have 3 to 5 minutes if you want, if we need it. Definitely, again, talk to a neighbor if you want to while you're doing this. So here's the setup. In this article that we've been reading from, every year around 150,000 babies in Nigeria are born with sickle cell disease, SCD. And 2006 study finds that 24% of the population are carriers. A carrier is another genotype term. It's an informal term, but what is a carrier genotype?
It can either be homozygous, that they have it, or heterozygous. They have one dominant and one non-dominant.
It's particularly the second one.
Yeah.
The carrier of a disease, we haven't talked about recessive diseases yet, but a carrier is somebody who has one mutant copy and one wild type copy of a gene. So what they're saying here is, in Nigeria, if you looked at 100 people, 24 of them, 24%, would have this genotype. They have a wild type version on one chromosome, they have the mutant version of HBB on their other homolog. And the prevalence of the sickle cell genotype, which is what genotype you have to get sickle cell anemia, which is minus over minus. So an individual that has both mutant versions of the gene gets sickle cell anemia, is about 20 per 1,000 births. So those are the data we need. 24% of the population are carriers, and the prevalence of the genotype is about 20 per 1,000 births. So the goal here in this worksheet is to fill in all the yellow. So start with the observed sickle cell disease frequency, that's one of these numbers up here, and then see in the next 5 minutes if you can get through the next 4, calculating the probability of 2 random Nigerians that might be interested in dating. What's the probability that they're both carriers? This is math that we've done before. This is if you reach into a random bag and you draw out 2 things, like you're looking for 2 adjacent nucleotides that are specific nucleotides, what's the frequency of any 2 Nigerians being carriers, and then go on down the rest of the questions on the Google Sheet. So I hope it'll get pretty noisy in here as we discuss and think about this. I will circulate and answer questions. About 5 minutes, we'll come back together. And especially, this part's not going to get recorded on the movie because now I'm going to be working on my laptop to do this demo. What was the easy thing to do? If there was an easiest part, what was the easiest part, or what's the first part that you tried to answer or enter?
Observed frequency is 20 divided by 1,000, which drop off the zeros, which is 2% of the population was observed.
So that was from it's a word question. It was from the word question. So remember, if you're in Google Sheets, everything that's a calculation starts with an equal sign, and then 20, that was the number of births of kids that have sickle cell disease in 1,000. So 0.02 or 2% of the population. What else is relatively easy? These two maybe seem deceptively difficult, but they're pretty straightforward.
The top one for the probability frequency of 2 random Nigerians being carriers, I just did the 0.24 multiplied by 0.24.
Okay, so that's the solution to the next. The probability of 2 random people being carriers. What's the probability that one person is a carrier if you picked at random? 24%. That was given to us in this magazine article. 24% carriers. So the probability of getting 1 carrier is equal to that number in cell C1. And the probability of picking anybody else that's also a carrier is the same frequency, which you multiply the probability of one person being a carrier with the same number. So it's 0.24 times 0.24. Just like the probability of having a C and a T next to each other in DNA is a quarter times a quarter, same calculation. Except this time it's the carrier frequency times the carrier frequency. So about, what is that? 5.7% of the time with this frequency of people that have one version of the sickle cell trait, about 5% of all combinations of people randomly, if they were just meeting randomly, dating randomly, having kids randomly, about 5.6% of the time. So for one of those 2 people, what's their genotype if they're a carrier?
Heterozygous.
They're heterozygous, which is, in our parlance, plus/minus. So if a plus slash minus person, if a heterozygote is making gametes, they've either got a plus, the wild type version to put in their gamete, or they've got the mutant version to put in their gamete. So what's the probability that a gamete has the capital S, which unfortunately I forgot to define for you, is the sickle cell, the mutant version? What's the probability that a gamete from a carrier is transmitting the mutation? 50%. Half of, right, you've got two alleles, one wild type, one mutant version. Half of the eggs are going to get, on average, half the eggs will get the wild type, half of the eggs will get the mutant version. Same with sperm. So the probability of a gamete from anybody that's a carrier is half, 0.5. Same is true for the other heterozygote that we've randomly picked out of the population, 5.76% of the time. So the frequency of person X and person Y having two S alleles, 50% of the time and 50% of the time, that's 25% of the time in 0.576% of the population, or 5.76% of the population. So the final answer is you multiply those three numbers. The probability that you pick at random two carriers, and then the probability that one of them passes the sickle cell gene into their gamete, and the probability that the other one passes the sickle cell gene into their gamete, sperm, egg. So 0.0576 times 0.5 times 0.5. The reason we're doing this is because we're going to be working a lot with allele frequencies and frequencies of things and making predictions. Most science is about making a prediction, testing it, then testing a hypothesis and seeing if your observation matches your expectation. So that's why over here, which you can't see, is labeled expectations. Because one of the goals of this class is making predictions and then seeing if your prediction is upheld by data. Yeah?
I did it a different way but got the same number, and it makes sense because it's the 0.5, but I used the Punnett square.
Yep.
And so if they have to just use it's the negative, which is 25%, multiplied by the top number. So is it okay if we do it either way? Does it matter to you?
It's the same logic, it's just a different mathematical exercise to get. So if you got the same answer and you used the same logic, then yes, totally fine. Okay, so expectation, frequency of sickle cell disease, we're predicting should be 0.0144. What was it? In the article they said there's a certain frequency. That's the carrier frequency, that's the frequency of the genotype, but
2%.
so we got pretty close, just based on probability. This means, in other words, that people basically are mating at random in Nigeria. What we'd expect, based on the carrier frequency, is you'd see about 1.44% of babies having sickle cell disease. They're reporting 2%. It's basically the same number. So we'll have other examples like this where I'll ask you to do those sorts of calculating expected values in the near future.
This does say 2 points underneath the Google Classroom thing.
Does it say 2 points? Well, we're not going to make it worth 2 points. Just because I said it, unfortunately. Okay. So we've already gotten through a lot of the vocabulary. We haven't started talking about PCR yet, which is going to take the next couple of classes after we get through dealing with alleles. So we've talked a little bit about distinguishing genotypes from haplotypes. We're going to talk a little bit more about haplotypes in just a minute. And the rest of these tasks are about PCR. We're going to talk a bit about DNA forensics too. So that's where we're heading. Now that we've got a little bit of vocabulary underneath our belts. This is probably, if you looked at this manuscript, Butler et al., Butler and others, from 2003, it's one of the most boring scientific papers you can read. Because what was it full of? for those of you that downloaded the PDF and looked at it.
Data,
Full of data in form of
pictures, or tables.
tables. Yeah, it's full of tables with numbers in it. That's, oh man, boring. The title of this, Allele Frequencies for 15 Autosomal STR Loci on U.S. Caucasian, African American, and Hispanic Populations. Basically what this paper is it's sort of the Bible of DNA fingerprinting. They're like crime scene analysis. You get DNA sample at a crime scene, you get a hair sample, a saliva sample, and you want to know something from that sample like which ethnic group, which race is the person most likely that left that DNA sample. So these tables are full of numbers are basically the guidelines for DNA forensics experts to try to figure out what race was the person that left this DNA sample. So over the next couple of classes, next time, we'll be talking more about, in detail, about how this is used and doing some more of these calculations to make predictions. So first, here's a figure of the 13 CODIS. I don't know if they still refer to this. There used to be a TV program called Law and Order. I don't know if it's still on TV or not. They were always talking about DNA fingerprinting in CODIS. It's a database that geneticists use to compare DNA samples. And this is showing the locations on human chromosomes of 13 different genes, 13 different regions, they're all genes, that they specifically genotype, they test to find out something about the ancestry or the race or ethnicity of crime scene samples. So scattered all across the genome, not all chromosomes, but some chromosomes. They're called STR loci. So what's an STR? One of the questions for class today. What does STR stand for? Maybe I should make this worth 2 points.
Short tandem.
Short tandem repeat. We've seen this in class before, but we just haven't called it STR. So STR stands for short tandem repeat. Okay, we're going to talk about the other two questions later, probably next class, because we need to talk a lot more about STRs and alleles before we get there. So short tandem repeats. We've seen them before. They're also microsatellites. STR, same thing as a microsatellite. We've seen microsatellites before. They're things like TA5 or TA7. So hopefully, if you see that notation, you'd be able to write out the actual DNA sequence underlying those abbreviations, right? 5 repeats of TA, 7 repeats of TA. These are not genes. These are just random sequences of DNA that exist in our chromosomes. But they're DNA. So these are two different alleles of part of your chromosome. So those might be at a specific spot on your chromosome 3, for example. Means that you inherited 5 repeats of TA from one of your parents, say mom, the maternal version, and your dad on his chromosomes had 7 copies of this repeat. So you inherited that other allele of this STR from your other parent. So that means, in this case, you're heterozygous for a size difference, the number of repeats at this STR on chromosome 3. So they're not genes, still alleles, different versions of a particular part of a chromosome. And we've already gone over the nomenclature bit of slashes, but we need to talk about haplotypes too. So that's where this comes in handy. Let's take a look at this individual, the offspring of 2 parents that we can't see right now, has 2 versions of chromosome 1. The 13, what does the 13 you think represent now that we're talking about STRs?
microsatellite number?
Yeah, the microsatellite number. This is all very relevant to this paper we're talking about, this genotyping paper that we're about to get into. DNA fingerprinting. So 13 repeats on chromosome 1 from dad, 14 repeats on chromosome 1 from mom. We can look at a different chromosome, and there's a STR on that chromosome too. So we can call this the chromosome 2 STR. Both of your parents gave you the same number of repeats at fertilization. So here on the left side is written the genotype, but we've added something now because we're talking about more than one chromosome. So the first example we started class with was just looking at one genotype of one gene, sickle cell, plus slash minus, minus slash minus. Now things have gotten more tricky because we're talking about chromosome 1 and its genotype and a different genotype of a different gene on chromosome 2. So we have to add another layer of complexity to the genotype, which is the semicolon, which separates different homologs. In other words, on chromosome 1, this individual is 13/14. It's 13 repeats on one homolog, 14 repeats on the other homolog. But then, when we switch chromosomes from chromosome 1 to chromosome 2, we have to add a semicolon, and then we do another genotype. Something/something. In this case, 15/15. And that's the exact same thing that was written up there already on this slide. We use slashes to separate the two homologs, the same chromosome, and then semicolons to separate different chromosomes from each other. Any questions? Slashes and semicolons. So let's look at the parents then. So for this individual down here to get a chromosome 13, chromosome 1, that has to be in one of the parents, right? Which parent was it in? Who gave the 13 copies on chromosome 1? Dad. This came from the gamete. So the sperm that made this individual, cartoon individual, the cartoon sperm that made the cartoon individual had a chromosome 13, or chromosome 1, 13 repeats of one STR, and on the other chromosome, 15 repeats. This is the concept of the haplotype that we're about to get into. We've looked at genotypes so far. That's looking at the two versions of the chromosome you've got in your somatic cells, like skin cell, brain cell, eye cell. The haplotype is defined as the combination of alleles that are present in the gamete. Haplotype because the gametes are haploid. So it's basically, another way to think about it is a haplotype is the DNA composition of a gamete. So in this case, the sperm has a haplotype. There's no slash because this is a haploid cell. So there are no homologs to separate. This has one copy of chromosome 1. It's got 13 repeats. So that's why its haplotype is 13 on one chromosome. We use a semicolon again to separate the two chromosomes. Then we tell the genotype, the haplotype, whatever allele is on, in this case, chromosome 2, which is 15 repeats. So haplotypes, genotypes. Genotypes talk about the composition of, in humans anyway, diploids, the composition of the same gene of two different alleles separated by a slash. When we talk about different chromosomes, we use the semicolon to separate them. Let's see how we did on Socrative. And then we're going to close by taking a look at what we need to do for next time, which is not nearly all of the stuff that's written on the last slide here. So wait just a second, I'll tell you exactly what we're going to do for next class. this last bit is more difficult than we've gotten to at this point. These first two questions, which were covered in some of the movies for today, but that's fine. We can do it in person as well. Question 1, for CT/GA, which is a haplotype? So what happens if we draw out, what does CT/GA look like if you drew out chromosome cartoons? Up to this point, we haven't looked at two letters next to each other without any slash or semicolon separating them. Okay, how many slashes are there? So how many chromosomes are in this individual?
One.
Or how many homologs?
One.
One homologous pair. So every slash separates two different chromosomes, the two homologs. Yep. So because there's one slash, that means there's two somethings that are being separated by the one slash. So we've got two chromosomes that are, say this is chromosome 14. Who knows? So that's the two slash. The CT goes on one chromosome. That means there's a C at one location and a T nearby it on the same piece of DNA. And when two nucleotides are not separated by any punctuation, no slashes, no semicolons, that means they're on the same physical piece of DNA. Not necessarily right next to each other, but somewhere on the same molecule. And then that indicates that there's a G and an A at the same spots in the same order on the two chromosomes. That is, if C comes first, when it's written CT/GA, then T comes second when you're reading across the DNA sequence. And that means that the G in position 1 on either side of the slash is paired with the C, and that means that the A, which is in the second position after the slash, is paired with the T. In other words, if I hadn't drawn the CT/GA and I asked you what's the genotype, let me number these. Let's say we're looking at position 1 and position 2. What's the genotype at position 1? We just write a genotype. What are the two alleles at gene 1? What was it? C, there's a C and there's a G. And there's a slash because we've got two different pieces of DNA. So that's the genotype at gene 1, G over C, G/C. And the genotype at 2 is T/A. So those are two genotypes. So the way that they get combined into a haplotype is the CT is a haplotype, which is a concept we haven't yet introduced. So it's a different color. That's a haplotype only because those two pieces, those two nucleotides came together on that piece of DNA from the gamete of one parent, because one whole chromosome comes from each parent at fertilization. To be a little bit more explicit, you could imagine if you go back in time, there's a sperm, there's an oocyte. The sperm had a chromosome that had a C and a T, so the haplotype of that sperm is CT. So it kind of looks like a frowny face, but that's not what I intended. And the oocyte had a chromosome that had a G and a C on it. So by themselves, each of those things is a haplotype, because it's a list of the genes that were in the genes that we're worried about at the moment, two nucleotides that were in a gamete. So when we write a genotype that contains haplotypes, what's on each side of the slash are the two nucleotides that are physically connected to each other on the same piece of DNA. CT, no separation, no punctuation between them, came from one gamete, and the GC came from the other gamete.
So it should be GA then.
Yes, thank you. I did that on purpose, I'm sure, to make sure that everyone was paying attention. There we go. CT and GA. The critical thing for this Socrative quiz to end, and then I'll talk last about what we're going to do for next class. The critical thing for this Socrative quiz then is to realize this fundamental point in genotyping, that something that looks like that is the same as C/A and G over T. Again, because the nucleotides that are listed first on both sides of the slash are the pair. Those are the same, that's gene 1, for example. And the things that are in the second position are, say, the 2 alleles at gene 2. So left-right order is important. So we've got genotype, multi-locus genotype here on the top, multi-locus, multi-gene. We've got single gene genotypes here at the bottom. So you should be able to, and we'll work on this, be able to interconvert between the two. For next class, now that people are trying to escape, which is totally legitimate because I'm out of time, don't do any of this, except for the bottom part. So this Canvas discussion's due tomorrow night, but we'll pick up where we left off and finish talking about DNA fingerprinting on Friday. Questions?